Today we're going to talk about RNA and how it compares to DNA, as well as the process of how these proteins are actually made. Okay? This process by which we go from DNA to RNA to protein is often referred to as the central dogma of biology. Okay? All right, some things to remind you of is we have genes, and genes are sections of DNA that code for the production of proteins. RNA is a single-stranded copy of that coded information for making those proteins. Okay. And looking at comparing these, so I have a structure or a picture of DNA and a picture of RNA. DNA is this double-stranded structure, and RNA over here is a single-stranded structure. And I'd like you to put these in your notes for sure and know all of these. The difference between RNA and DNA is that the sugar is different. So instead of using deoxyribose, RNA uses just ribose. RNA is single-stranded versus DNA that is double-stranded. And then RNA contains a nitrogen base called uracil instead of thiamine. So if you're looking at a strand of nitrogen bases and you have ACUG, it's going to be RNA because U's or uracil is only found in RNA, and T's or thiamines are only going to be found in DNA. There are three different types of RNA. There is messenger RNA over here, which we call mRNA, and the one we'll probably talk the most about. The next one is our ribosomal RNA, often called rRNA, and that is shown here, and this is going to actually help code for the ribosomes themselves. And then you have a thing called tRNA or transfer RNA, which we'll talk also about. The first one are messenger RNA. And what messenger RNA does is it carries copies of the instructions for assembling amino acids to the proteins. And here you have within our nucleus, and this is showing the nucleus, what has happened here is mRNA has made a RNA copy of DNA as far as creating the instructions for DNA. So mRNA will come in and it will create a RNA copy of DNA, and then the mRNA will leave and it will go to the ribosome, where we know that proteins are actually made. Our second type of RNA is this rRNA, and basically it helps make up and code for part of the ribosomes. Do you recall where ribosomes were made? Please put that answer in your notes. Okay. Our next type of RNA is called tRNA, and it's our transfer RNA. And what tRNA does is tRNA brings the amino acid to the ribosome in order for it to be linked into these chains, these polypeptide chains, which make up our proteins. And this is the structure of a tRNA molecule. And these are three RNA nitrogen bases that are attached to this amino acid. The process of protein synthesis has a couple of different steps. So we go from our DNA strand, which is our template strand, and then a process called transcription occurs. With transcription, we make an mRNA or a messenger RNA copy of the DNA. Things you will notice here is you have C's with G's still, but instead of A's going with T's, you have A's that are now pairing up with U's because there are no T's in RNA. You'll see no T's. Here is the DNA copy, so you see a T, and the corresponding base for that is A. And then with A, you now have U instead of T. From mRNA, we go through a process called translation, and translation is how we actually make the proteins. And so this is these amino acids being brought over based on the RNA code that is given. Okay, so some things about transcription. This is a DNA copy, which is made in the form of RNA. So in transcription, it's our first step when we go from DNA to RNA, and more specifically mRNA, which is our messenger RNA. This begins at the section of a gene that's called a promoter, similar to what we saw in DNA replication. Okay. How do you think transcription differs between eukaryotes and prokaryotes in the location that it occurs? Please answer this question in your notes as well. Okay. Some things about how the RNA is actually processed. So much of the DNA within the gene is not needed to make a protein. We do have a lot of excess DNA, and these areas are called introns. So DNA that is not needed is called introns. The part that actually codes for protein is called exons. So here we're showing DNA and goes through transcription, and there is a pre-mRNA copy made. And here we get just these exons because that's all we actually need to make our proteins. So if you think of exons as expressed and introns as in between. Okay, so the introns actually get cut out of the RNA molecule, and the exons get spliced together to make mRNA. Some things about the genetic code. It is a universal code for all of the organisms and all of the living things in Earth. And basically, we can use our mRNA and decode it to figure out what amino acids are needed. A codon is the name of 3 consecutive nucleotides that code for an amino acid. So we can read these 3 at a time. So we have 1, 2, 3, this is a codon, 1, 2, 3, this is a codon. So our codon here is AUG, and our codon here is AAC. We have these things that are called codon charts, and this is how we're going to be able to decode this mRNA, and yes, we use only mRNA on these charts to figure out what amino acids there are. So you have your DNA, you go through a process called transcription to get your mRNA copy, and then translation is when we actually figure out what amino acid is where. So we have a specific codon, AUG, and how you would find AUG is you start in the center, and so we would find A, and then you work your way out, and we go U, and we go G. And what's special about AUG, which is methionine, that is our start codon. So this signals to start making a new copy of the protein. So start a new chain of amino acids. So methionine is how it starts. And if we were looking up something like UAU, we would go to UAU, and we would have tyrosine. And if you look around the outside, you may see that some of these amino acids have multiple ways that you could get the same amino acid. So you would have something like GCU and GCC are all of the code for the same amino acid, which is alanine. Which is a good thing because if you have a mutation where you have an A in place of a C where it originally would be, you still get the same amino acid. You may notice around the outside here that you have things that are called that have the word stop next to them. These signal you to stop making the protein, that the protein is complete. So you could have UAA, and that would also signal the protein to stop. One thing you need to write into your notes is not this, you will be given a copy of this, but you should write that AUG is the start codon. There are other codon charts that you may see, and they work a little bit differently. This is a little bit difficult to see, and I don't have it zoomed in, but basically you can follow the directions on it. So this is have your first letter of your codon. So you would find like A, and then your second letter would be up here, so C. So you would kind of find it like a graph. You would go A, and you would go C, and then you would find your third letter, which could be U, and you would have ACU, which would be like tyrosine. So, and that process of translation that we discussed is when you decode mRNA into a polypeptide chain. So translation is used by that codon wheel that I just showed you. And translation is going to happen on the ribosomes. Transcription happens in the nucleus, and translation happens on the ribosomes. Because our ribosomes are where proteins are made, and translation is how we're going to make these proteins. Okay, so the ribosome binds a new tRNA molecule and the amino acid as it moves along the mRNA. So here's our section of mRNA, which was made in the nucleus, and it was taken to or traveled to the ribosome. Here we have these tRNA molecules, which stand for our transfer RNA, and they're coming along with the corresponding RNA and laying it down and forming a polypeptide chain. So if you see here, we have A going with U, A with U, G with C. Our start codon is AUG, which is methionine, and the anticodon helps find this because A and U are attracted to each other, and G and C are attracted to each other. And it's bringing over methionine, which is our start codon. So if we were to look at that, if we were to go back and look at that chart, we would know that AUG is methionine. And then our next one we would look up our mRNA, which is UUC. We could go back and we would say UUC. And we have an amino acid that is called phenylalanine. And you would see up here on this transfer RNA molecule, they are carrying phenylalanine. Here's our other tRNA molecule, and we have AAA, and it is bringing lysine. So if we looked up lysine on that chart, we would see that AAA was the mRNA structure that coded for lysine. Okay. So this is showing this protein synthesis occurring. And as you can see, so this one, the methionine one is leaving, and it is joining these together. So you start to get these long chains of amino acids that are joined together in the ribosome, thanks to these tRNA molecules bringing over the correct amino acid. Okay, and this process will continue until we see one of those stop codons. And remember, these stop codons were here. Okay, so it would go all the way along, and we have our polypeptide chain, which is made of amino acids. So this is making